another A-level biology video of Mr Phillips. Today, going to be revising the structure of prokaryotic cells and viruses as part of the year 1 of the A-level course. So let's start off then. So this basically two pictures that three pictures you should know: a prokaryotic cell and two eukaryotic cells. So just to revise with you really briefly the structure of an animal cell. Okay, so we'll just go through some labels. You should be able to label the ultrastructure of an animal cell. Remember, the ultrastructure is the structure you can see under an electron microscope, which will come up in a follow-on video shortly. So you've got rough endoplasmic reticulum, so that is a membrane covered in ribosomes. You should know ribosomes are involved in protein synthesis. You've got the mitochondrion, which is obviously involved in respiration, the aerobic stage of respiration. You've got ribosomes that are free in the cell, also involved in protein synthesis. You've got the nucleolus, which is involved in making the ribosomes. You've got the nuclear membrane, which controls what enters and leaves the nucleus. You've got the Golgi apparatus, which I'm just covering up at the moment, which involves in packaging and repackaging and modifying proteins. You've got the centrioles. We won't talk about centrioles too much at the moment, but they're involved in cell division. Smooth endoplasmic reticulum, which is just membranes which are involved in the production of lipids such as steroid hormones. And you've got lysosomes, which are membrane sacs containing hydrolytic enzymes that can break down old parts of the cell and can break down bacteria invading cells. So we need to look more today then at prokaryotic cells. The word prokaryotic means before the nucleus. And what that means is referring to basically primitive cells that do not have what we call a true nucleus. And I always put that word true nucleus in there, so we mean something that doesn't just look like a nucleus, but it's actually a nucleus. The eukaryotic cells obviously do have a true nucleus, and they have membrane-bounded organelles such as mitochondria. Prokaryotic cells don't have complicated membrane-bounded organelles like mitochondria. So you need to be familiar, or very familiar, you need to draw out and label and explain the roles of the different parts of a bacterial cell. So you've got an electron micrograph at the side of a bacterial cell. I can't see a flagellum on that one, but obviously we've got a flagellum at the bottom of the sort of cartoon version. So if we just go through then what sort of things we have which are similar and different. Okay, so you've got a cell wall. So you can see the cell wall going around the cell. The thicker layer just on the outside of the cell here. Okay, cell wall again is involved in structure of the bacterial cell, just as in a plant cell. The only difference is instead of cellulose, it's made out of a polymer called murium, which we'll talk about in a few moments. Okay, then you've got the cell membrane, which is just beyond that sort of line as well on the inside, and that again controls what enters and leaves the cell. Now, the structure of the cell membrane, the cell plasma membrane, or the cell surface membrane is identical. So it's also a phospholipid bilayer, as well as in eukaryotic cells. Okay, we have inwards foldings of the membrane. So you don't have organelles like mitochondria, but you have membrane. The outer membrane, the cell surface membrane, does fold in to form networks of membrane, and those are called mesosomes. Now, why they're quite useful is that's a good place if you haven't got organelles for your enzymes to attach to. So respiration largely occurs in bacterial cells on mesosomes, which is quite important. Okay, you then have a capsule or a slime layer. Okay, and the capsule or slime layer is obviously it's got its not every single one of them has a capsule or a slime layer, but it's just basically to protect the cell from attack from the immune system of whatever it's invading. We've got a plasmid. Okay, the plasmid is a small circular bit of DNA that is found in bacterial cells, and it gives them antibiotic resistance, because it's much more important in year 13. You've got a nucleoid, which is basically a singular chromosome. So the DNA in bacteria or in prokaryotes is a singular chromosome, and the DNA is not associated with histone protein as it is in eukaryotic cells, so therefore we call it naked DNA because it's just floating in the cytoplasm. And it's also, so there's two factors: it's not in a nucleus and it is naked DNA, which means it's not associated with histone. You've obviously got a cytoplasm. The ribosomes, which contain, do protein synthesis, obviously needed. We need proteins in bacterial cells as well. they are smaller and they're called 70S, which means I think 70 subunit ribosomes. And then we've got a flagellum. Not all bacteria cells have flagellum. Some bacteria cells have multiple flagellum. A couple of animal cells do. So think about a sperm cell, the tail's proper name is a flagellum, and they are basically a whip-like structure. It rotates to cause the cell to be able to move where it wants to. So let's have a look then. Some extra information on organelles then. So we need to know just a couple of bits of key information. So the bacteria cell wall has a basic structure of a mesh of protein called murein. That is just, again, strong protein like cellulose is for plant cells, but obviously they're not the same as plant cells. Plasma membrane is the same structure in both eukaryotics and prokaryotics, as I said before. And then we need to know that prokaryotic cells do not contain membrane-bounded organelles, so they don't have chloroplasts, they don't have mitochondria, but they do have these infoldings of the membrane called mesosomes, which allows the enzymes for respiration to be attached. So they don't have the same respiration as we would as animals or plant cells would have, but they do have a more complex one than just anaerobic respiration. And the genetic material, as we said, is packaged into a large singular DNA bacterial chromosome. It's not in a nucleus, and it's not associated with histone proteins, so nuclear membrane. The ribosomes are present still, they're still in protein synthesis, but they are smaller than in eukaryotic cells. That's the key bit there. So bacterial tail is a flagellum, and its structure rotates and helps it. Right, so you could be asked quite easily in a multiple choice question or in a just a simple question or even a bigger question to describe the structure of prokaryotic cells or eukaryotic cells. So prokaryotic cells, such as bacteria, have cell membranes. They don't have a true nucleus. They have no membrane-bounded organelles. They have a cytoplasm, and they belong to the kingdom bacteria. Eukaryotic cells, however, do also have a cell membrane. They have a true nucleus, so membrane-bounded, or have a nuclear envelope. They have membrane-bounded organelles such as mitochondria and chloroplasts. They've got a cytoplasm, and eukaryotic cells contained in the kingdoms animal, plants, protists, and fungus. So protists and fungus key there are eukaryotic cells. Just to do a bit more comparison, I mean, it's a bit more important to get these comparisons in. So the size of the cells are different. So you see a difference in sizes in micrometers there of the cells. But as we said, eukaryotic cells have circular DNA, whereas we don't have circular DNA in eukaryotics. Prokaryotic cells, sorry, have circular DNA. We don't as eukaryotics. They have no nucleus, there's no nuclear membrane, whereas the eukaryotes do have a nuclear membrane. The DNA is naked, and just to make sure make sure you know what that means it is not associated with histone proteins. So our chromosomes are made up of DNA and histone proteins. The DNA is just DNA floating in the cell in the cytoplasm in prokaryotes. The ribosomes are smaller, so they're 18 nanometers in diameter, whereas they're 22 in eukaryotic cells. There is no endoplasmic reticulum present in prokaryotic cells. Very few simplified organelles. There is always a cell wall, though, where it's made of murein or anything else, whereas in eukaryotes, obviously, only sometimes it's found in fungus and plant cells, not in animals. You need to know a bit about how prokaryotic cells reproduce, okay? So effectively, the cell cycle, which is something you'll study later on, is key in all living things because it's how cells reproduce. But basically, compared to humans, when you look at mitosis, you look at the details of, not humans, so in animals, you look at the details of it. It's a lot simpler because they've only got single strands of DNA in prokaryotes. The plasmids also get copied, and it's done by something called binary fission. So why is it simpler? First of all, there's no organelles to copy, okay? Because we don't have those organelles. The cytoplasm just initially breaks down in half, and then it can grow afterwards. And there's no nuclear envelope to break down, okay? There are no spindles, but there are mechanisms to ensure that only one set of DNA goes into each cell. So you don't need to know that detail, but it does happen. So if we have a look at what happens then in the cells, we sort of stage the binary fission in different stages. The first thing that happens is your DNA replicates, okay? So DNA goes from 1 to 2, the DNA is replicating. Now that is the main nucleoid, the main bit DNA, which replicates, and you get one copy of that. But also, if you had plasmids in there, the plasmids, which have our DNA, are also going to replicate. Now, whereas the nucleoids only replicate once and only ever make like a copy of themselves, the plasmids can make as many copies of themselves as they want. The cell then grows and it gets bigger. Okay, so it grows and it starts replicating things that are needed for it. And the DNA, just like in mitosis, moves to the poles, to opposite poles. And then the cell undergoes cytokinesis. And basically what happens there is you can see this infolding of the membrane, splitting the bacterial cell into two. The cell then splits up into two daughter cells, and they're identical copies of each other. Each one will have one copy of the circular main piece of DNA, but they'll have variable numbers of plasmids. It doesn't matter because the plasmid's job is the antibiotic resistance. So you might have one plasmid over there, two plasmids on this daughter cell here. It's not important as long as you've got the same identical copy of the main piece of DNA in there. Okay, so a virus is non-living, okay, but we obviously need to know a bit about it. So it obviously is really important because obviously with the coronavirus, it's obviously had a massive impact on the whole of human civilization. Viral studies are so vital in human civilization, effectively. So what do we need to know? We need to know a bit about the structure of the virus. So if you have a look at the picture at the top of this slide. Then this is showing you a particular type of virus. I think this is a bacteriophage, which is a virus that infects bacteria. And we're actually useful at those in genetic engineering and things like this. So this one sort of shows you the structure of the virus is that you've got a DNA molecule here, okay, and the DNA molecule is, and it can be DNA or it can be RNA, is inside a protein capsule or protein capsid, as it's probably more likely to be called. And what will happen is on the outside of that, there will be proteins, and lots of different formats. So on different viral cells, but there are proteins that stick out and they attach to the host cells. And viruses will have proteins sticking out of the host cells that are complementary to the cell that they are invading. And basically, they'll have a method of injecting the bacteria into the host cell, and then that goes on to viral reproduction. So viral replication is obviously quite important. It's obviously how viruses spread in our body, particularly important at the moment. So what we need to know is how they do it. Well, basically the virus comes into the cell. The virus can't replicate on its own. It has to be inside a host cell. So you can see on this very small virus at the top here, the sort of green proteins sticking out, which are the receptor proteins we talked about before. And the receptor proteins, the virus proteins, will be complementary to receptors on the host cell. Once they've attached to the receptors, which are on the cell surface membrane of the host cell, they'll then inject the viral DNA into the cell, or the viral nucleic acid, I should say, because it could be RNA or DNA. Okay, and then the virus, then its RNA or its DNA will incorporate into the DNA of the host cell using an enzyme called reverse transcriptase. And then when the cell replicates, when it replicates its DNA, it will replicate the viral nucleic acid as well. And then what will happen over time, depending on length of time, will depend on what the virus is the virus will they'll make new virus parts using that DNA in the cell to make new viruses. And then eventually the virus will get to a stage where it's no longer sort of what we call the latent stage, where it will get enough viruses or it bursts out into the cell as a new virus. Now that virus then will come and attack other cells. So the key thing that you need to know is the virus has proteins on its coat which recognize receptor proteins on our cell surface membranes. It injects its nucleic acid into our DNA. When the DNA in our cells replicates, it replicates the viral DNA, and then the viral DNA replicates to make new viruses with our cells, and then the viruses burst out of our cells and go and invade new cells. So that's been useful. I'm Mr Phillips, and I'll see you again soon.